truly great pleasure to it's my truly great pleasure to introduce Sarah Burlage, coming from here at Dana-Farber. Sarah did her undergraduate degree at Miami University of Ohio, moved a little north to the University of Michigan to get her PhD, and then came to Boston to the Broad Institute and Stuart Schreiber's lab to do postdoctoral work. When I was looking down Sarah's CV for many honors and good things to say, one thing really stood out to me, and it's a comparison that I can make basically because I'm so old. And that is this. When Sarah came here around 9 years ago, she set out to kind of do what it says up there on the slides, set up a system for a whole new class of enzymes, the DUBs. There are about 100 of them. She'll tell you all about them. But I'm old enough that I'd gotten to sit in at Ciba-Geigy when a group of people there set out to make kinase inhibitors. And the problems were remarkably similar. Not much structure, very, very little. No chemical scaffolds, no libraries of large chemical matter that could be screened. No omic, no dubomic, no kinomic screens. The literature, that was something I was supposed to be taking care of, wasn't all that good either, frankly. Of course, Alex Matter, Nick Leiden at Ciba-Geigy, now Novartis, had a small army of folks and tens of millions of dollars to throw at this problem, and they ultimately succeeded. When Sarah came here, she had her startup package and herself. Now, we were just talking about startup packages. I was her department chair then, and I really wish her startup package had been competitive with Alex, but it really wasn't. But as you're going to see, Sarah and her lab and her colleagues have just done amazing things. I personally find it incredible, particularly thinking about this comparison. So at that point, I'm going to shut up and we should welcome Sarah.
All right, well, good afternoon, and thank you, Tom, for the introduction. As I go through some of these intro slides, I swear I did not tell Tom what I was going to be saying, but thank you for kind of priming for the intro and some of the analogies that I was going to make. But I'm really excited to be able to update some colleagues on the work that we've done and introduce it to others, really around these key regulators of ubiquitin, the deubiquitinating enzymes. And so before I talk about the deubiquitinating enzymes, I just wanted to take a step back and really talk about what it was that led me to be interested in the DUBs. And so really throughout graduate school and postdoc, what I was really passionate about was trying to go after these very difficult to drug targets, transcription factors in graduate school, protein-protein interactions when I was a postdoc. And part of the reason I was really drawn to these types of targets was because it gave you the real opportunity to think about innovative strategies, approaches, new modalities, to gain access. And I really enjoyed that chemical challenge. So after a couple, years of postdoc over at the Broad, I intended to go, work in industry, be a medicinal chemist there. But I had a unique opportunity to come over to Dana-Farber. So actually my first role here was as, the first staff scientist in the newly formed medicinal chemistry core. In that group, I worked very closely with our now very missed colleague, Nathaniel Gray, to establish and grow that group. And there I worked on a very different set of targets, working on kinases. Previously, very underexplored, but by the time I started working on them, there was a nice established roadmap for, the types of compounds that target them. There were a lot of resources in terms of libraries, in terms of assays, And so for a while, this was a really nice change for me. But after a few years, I really had that yearning to go back to an early stage understudied target class. Really having, an opportunity to be able to illuminate how do we go after these? How do we target them? And then what do they do? What are they important for, in a cell and, in disease biology? So as I was thinking about where to focus, I thought a lot about the ubiquitin system. Right, I think especially, 10 years ago, we were just starting to appreciate that different components of the ubiquitin system are, really critical in normal physiology and human disease, but we didn't know a lot of details. Most of the enzymes, most of the proteins involved were really understudied, and we had very few chemical tools really to go after these. So this post-translational modification of ubiquitination, it is regulated by a cascade of ubiquitin activating, conjugating, and ligating enzymes that attach ubiquitin to a substrate protein. And one of the effects that this can have, the one I'm showing here, is then tagging it for degradation by the proteasome. But of course, there's a lot of non-degradative ubiquitin in the cell as well. And then ubiquitination can be reversed by the deubiquitinating enzymes or DUBs. So then you can think about being able to inhibit a given DUB to promote degradation of its endogenous substrates. So this would really then kind of be in contrast or complementary to a modality many of you may be familiar with, which are the PROTACs, where bivalent compounds will hijack an E3 ligase, drag it to a non-natural substrate of the ligase to be able to promote its degradation. And so for reasons I'll kind of outline for you on the next couple slides, I was really drawn to the deubiquitinating enzymes as an area to focus. So this is a family of 100 enzymes spread across 7 families. With the largest being ubiquitin-specific proteases, or the USPs, which is the family shown here in green. So most of the DUBs are cysteine proteases, so they have kind of a standard catalytic triad that you will find in other families of cysteine proteases. And the DUBs are referred to really kind of as a hand. You have the fingers, which will coordinate ubiquitin, and direct the C-terminus into the active site where the catalytic cysteine will cleave the isopeptide bond that links ubiquitin to its substrate protein. We know from structural studies that the DUB catalytic sites, they all have the same confirmed conserved catalytic triad. And then within families, there's a common fold but limited sequence homology. And then we see divergent architectures across families. So that really added up to me to suggest that this should be a druggable family of enzymes. And then, DUBs typically aren't just catalytic domains in isolation, but rather, large multi-domain proteins, such as what I'm showing here for USP7. And these, additional domains really are important for substrate recruitment through forming protein-protein interactions, for example, as well as regulating catalytic activity. So thinking about the biology of DUBs at the time, again, I've noted they were understudied, but I really felt like there was enough out there to convince me that these are going to be, very critical in various cell functions. So for example, we could see there were multiple DUBs that associate with the proteasome or with, different chromatin remodeling complexes. And sequencing studies had identified DUBs that had both gain or loss of function mutations. And then perhaps a little bit, less rigorously characterized were some relationships between DUBs and different oncogenic proteins. But at least raising the possibility you can inhibit a DUB like USP28 to drive degradation of the transcription factor, MYC. So it really felt like if we could start to get some chemical tools, there should be tremendous opportunity to contribute to the understanding of individual DUBs, as well as the enzyme family as a whole. So, I ultimately managed to convince myself to make this jump from this nice, well-established world of kinases that I was working in, where they're clinically validated, you have a lot of resources, you have a lot of colleagues working in the space, and moving into this really uncharted territory of DUBs. And I can thank Tom for really, already highlighting these points. I'm not exaggerating here. When we jumped into the space, there were really no potent and selective inhibitors, no DUB ligand co-structures. And without these two things, certainly, we don't know how to make DUB-focused libraries, which is an asset that can just be so enabling for, an enzyme family. And again, very functionally understudied. So you can probably appreciate then this, real mix of enthusiasm for what we might be able to accomplish as I was starting the lab, but really balanced by, many, many sleepless nights worrying that what if none of this can actually be accomplished? And so fortunately, the dub field looks very different now than it did, even 8 years ago. There's been significant progress in developing selective inhibitors, understanding the function of individual dubs, as well as development of new enabling technologies. And I'll be sharing with you today some of the ways in which my lab has contributed in these areas. But I also on this slide just want to point out I think a really important milestone for the field that's occurred in the last 18 months, and that's kind of the first of the modern dub inhibitor clinical trials. So KSQ Therapeutics has put agents targeting USP1 into the clinic. These are being investigated in advanced solid tumors, in particular those that are BRCA mutant alone and in combination with olaparib. And then Mission Therapeutics, which is a dub platform company, has now advanced 2 USP30 inhibitors into the clinic, the first for chronic kidney disease and the second for Parkinson's disease. And these have now actually advanced into phase 2, so cautiously optimistic that we will have the first approved dub inhibitors here in the next few years. So what I want to talk about today is first just a quick introduction to how we've thought about building out a target class approach to really go after this enzyme family in the lab. But I'll primarily just exemplify our approaches, with a couple examples. And then at the end, talk about, some new early stage work that we're excited about that starts to move beyond protein degradation and get into the area of protein stabilization. So as we were getting started, right, we're a group of chemists. We really want chemical tools to interrogate this enzyme family. So getting chemical matter was the highest priority. And we knew where we ultimately wanted to end up, and where we ultimately wanted to be was having rationally designed focused libraries. These libraries purpose-built for dubs, really built around cores that bias to this enzyme family over other protein families in the cell. And we've heard how this has just been so enabling for kinases, and that's what we ultimately wanted to be able to do. But we didn't have anything to start with, so we really needed to just focus by getting our initial chemical starting points, finding pockets, and generally building knowledge, around targetability before we were going to be able to have these libraries, which is, where we wanted to get to. So we've done a lot of different strategies and approaches. I'm just going to tell you about 2 that, really have been beneficial for us. The first is pretty simple: optimization of known compounds. You can sometimes find things that are fairly mediocre in the literature, but you can work with them really to get high-quality compounds. And in fact, the story I'll tell you about today, we did that, and this ultimately led us to our first dub-ligand co-structures and really proof of concept that you can, in fact, get potent and selective inhibitors for this enzyme family. And then second, we really had to do a lot of screening. Any of you that have done screening appreciate that this is tedious. And it's a lot of work just to get a starting point. But when you don't have any starting points, this is simply, the stage at which, you have to start. And then, of course, it's not just about getting inhibitors. What's really important is then being able to put these to work to understand function of dubs. So of course, then we can take individual compounds in a target-specific manner. For dubs, which there is suggested biology, pharmacologically investigate this. But where I think our platform really is uniquely enabling is in this area of chemical genetic screening, where now you can investigate in a target-agnostic manner, so not focusing on an individual dub, but across the enzyme family, to identify novel dub function through these, chemical genetic screens. And I'll share some examples of where this has been very successful today. Okay, so when we are getting started, right, the first thing you really need is a project to start to build capabilities and knowledge base. And for us, that first project ended up being USP7. I talked about utilizing known inhibitors. We looked at everything in the peer-reviewed and patent literature, and there was only one compound that really struck me as drug-like. And that was this hydroxypiperidine-based compound 1. So we made this compound, we confirmed and inhibited USP7. It was pretty weak. But we saw that it was very selective, and there hadn't been any selectivity data reported in the patent. So this was really exciting for us. Definitely having to improve potency over selectivity is the place where I'd like to be. So we were able to implement these general dub assays that can be applied for, the majority of different dubs to drive the optimization of this particular chemical scaffold. A basic dub enzyme assay is simply looking at the ability of a dub to cleave the fluorogenic substrate, ubiquitin rhodamine. And then we can look at target engagement, confirm we're binding the dub in cells, utilizing these activity-based probes that will covalently tag the catalytic cysteine of a dub unless it has been blocked by your compound. And you can use either Western blot or mass spec to read out this cellular target engagement. Okay, so this slide summarizes several years and 200 or so compounds of work that really led us to a very high quality chemical probe for USP7. We're able to get from our 12 micromolar starting point down to this XL177A compound, which is a covalent inhibitor of USP7 that actually has subnanomolar inhibition of the enzyme, and it can be paired with its enantiomer as a negative control. Now, along the way, we published a first-generation probe. It's XL188. You're looking at the co-structure of this compound bound to USP7. And while it seems like it should be pretty good as a 90 nanomolar inhibitor, it wasn't great in cells. And we've seen this multiple times now, perhaps due to the high concentrations of ubiquitin that are in the cellular environment. So we thought that perhaps we could optimize this first-generation probe to improve potency by making it covalent, targeting this catalytic cysteine. And moreover, this would then allow us to address a question that we had, which is, are we going to be able to target this invariant catalytic cysteine while maintaining high selectivity for a dub? And so ultimately, we landed at this compound that has this very kind of unique tetrahydroacridine warhead. As I've already said, very potent, but what we are very pleased to see, also very selective. And we can see that when we measure selectivity across 3 independent experiments, looking at purified dubs, native dubs using competitive activity-based protein profiling coupled with quantitative mass spectrometry, and then even looking across the entire proteome. So we were really happy then to now have high-quality chemical matter to start to look at the cellular impact of inhibiting dubs, and in this case USP7. So we've now done a lot with these compounds in our lab through collaboration. So I just want to share 2 brief stories with you today. And both of these were done in collaboration with colleagues here at Dana-Farber, and both initiated when they had USP7 as a top hit in one of their genome-wide CRISPR screens. So the first was a collaboration with Kim Stegmeyer. And so as she was, working with, investigators at the Broad and St. Jude to generate the pediatric cancer dependency map, she was really struck by what a strong dependency USP7 was across a number of different pediatric malignancies, and that this dependency was really specific to cancer cell lines that had functional P53. So she, of course, went on to validate this genetically. She didn't know that at the time that we had nice USP7 inhibitors we were working on in the lab, so she got the one that you could buy from SelectChem, and it didn't phenocopy her results. So this was leading to a lot of frustration. I can tell you that compound, it's pretty multi-targeted, and that really turned out to be the problem because when she then got our inhibitor, it nicely phenocopied the genetic results. The wild-type tumors were sensitive to our compound, the mutant ones were not, and then you could CRISPR out P53 and reverse the viability effect, confirming that this axis is important for the observed effects. You see the same thing now when you go to the adult cancer cell lines, where the P53 wild-type lines are more sensitive to USP7 knockout than the adults. And that is both with genetic knockout as well as profiling our compound against a large panel of cancer cell lines, leveraging the PRISM technology over at the Broad. And so this was really exciting when we got this PRISM data back, where they look at the profile of your compound and compare it to the profile of all the CRISPR knockouts they've done in the same set of cell lines. And what our compound looked most similar to was USP7 CRISPR knockout. So it's on target. There's no question about it. And then furthermore, it looked very similar to MDM2 knockout, anti-correlated with P53, and we could see that a mutation in P53 predicts for insensitivity to USP7. I will, just kind of point out then, the other really nice thing that we can learn from, these studies are what are some of the most sensitive lineages to USP7 inhibition. And they might not be what you would predict. In the adult malignancies, it's renal cell carcinoma and melanoma. And so now an interesting area of investigation is really trying to understand, why those particular lineages. And so how does USP7, affect these P53 wild type tumors? And really what it's doing, inhibition of USP7 is ultimately leading to upregulation of P53, probably what you're predicting was going to be the case. And it does this through modulating a number of the negative regulators of P53. So you inhibit USP7, you promote degradation of a number of these ligases as well as an E2 enzyme that work to stabilize P53. So this differentiates us a bit then from, MDM2 inhibitors, which are going to be modulating, just one of these negative regulators of P53. And then, really consistent with what we'd seen thus far with regulation of this axis being a dominant activity of USP7, when we do transcriptomics, we see a really strong P53 gene signature. So that then takes us to the second study that ended up, being a completely different area, of biology that you don't observe when you're looking at viability really as the fitness metric. And so this was a collaboration with Kathy Wu and Jim DiCaprio. So they did a genome-wide screen. What they were looking for is genes that when depleted would increase HLA on the surface of Merkel cell carcinoma so that you could get increased antigen presentation to T cells. And they were focusing really in Merkel cell carcinoma because HLA is transcriptionally downregulated in this particular malignancy. And when they did their screen, they had USP7 as the top hit. This was really interesting. There really hadn't been any, inclination in the literature linking USP7 to this particular phenotype. So they, called me up. We were very happy to be able to share our compounds and nicely saw that pharmacological inhibition of USP7 increased HLA on the surface of these cell lines, also led to increased abundance of displayed peptides. In further control experiments, you can see that this effect is USP7 dependent, P53 independent, but I guess what I neglected to highlight on this previous slide was some of these other, key genes that were, having a similar impact were in this PRC1.1 complex, which USP7, there was a little bit of data suggesting it could reside in that complex. But here we see it's only partially dependent. So there's really more mechanistic work to do. And importantly, we now have compounds that can be used in mouse studies to see if this ultimately does lead to immune surveillance. Okay. So that's where I'm going to stop with the USP7, project for today. But just, highlight, this has been a really impactful target for us to work on. It provided proof of concept for potent and selective covalent and non-covalent inhibitors. It got us our first co-structures. We are able to then, use these chemical tools to pharmacologically validate different CRISPR data showing this viability dependency in P53 wild-type tumors, as well as regulation of HLA on the surface of tumor cells. So we're really intrigued now to further investigate as we go forward the potential of a USP7 targeting agent to directly kill cells due to upregulation of P53, as well as, really sensitize to immune surveillance. In addition to some of those, translational type questions, also digging in now to some more mechanistic questions that we can now pose with this original generated knowledge base. But of course, we haven't set out to be a USP7 lab. We set out to be a dub lab. And so we need more compounds that are targeting dubs. And, we are really fortunate to be able to establish a great collaboration with Novartis Institute for Biomedical Research to screen dubs. So I told you at the beginning where we wanted to get to was a library with dub-focused compounds. So just to be clear, that's not what this was. This was just a diversity set that they had developed, that they had assembled there at Novartis, but we hoped that by screening it, we'd gain insight into, chemotypes and pockets that might ultimately get us, to our dub-focused collection. So we screened their non-proprietary deck against 8 dubs using this, general dub enzyme assay that I've already introduced. And so here's just a high-level summary of the results. So kind of, each of the pie pieces here are outlining for you the number of hits for a given dub, where we have in the outermost ring the total number of hits. So for USP7, that was 53. And then as we apply increasingly stringent criteria for potency and selectivity, we whittle it down to our favorite compounds in this innermost ring. And so, of course, what's very striking when you look at the innermost ring is that we have a very large number of hits for USP28, but no other dubs. So we picked a handful of these top hits, resynthesized them, validated them, and really confirmed inhibition of USP28 with potencies from a couple hundred nanomolar up to, the low micromolar range. And then when we assessed selectivity against a broader panel, we saw overall excellent selectivity, although they were also inhibiting USP25, which is nearly identical within the dub domain. Now, kind of interestingly, while we were doing our screen, others were also looking at USP28 and independently found a very similar scaffold that was reported in a patent, this compound B55. And so, when we had that initial screening data, we were just so curious. Why is this such a targetable dub? And so, we wanted to pursue structural studies with these very inhibitors to see if we could find, new pockets that might lend insight for USP28 specifically, but hopefully then extend to other members of the enzyme family. And so working with Dana-Farber's structural biology core, we were able to obtain a co-structure which showed the compound, this particular compound, B55, which was similar to one of our other hits, kind of bound in this deep pocket, formed, kind of at the intersection of the thumb, fingers, and palm, making extensive interactions with this alpha-5 helix, as well as this kind of beta sheet that makes up the palm. But just big picture, where is this? It's now in a new pocket that we hadn't observed previously in our, our structural studies and our work with USP7. In this distal pocket that's further away from the catalytic cysteine, the compound is more than 10 angstroms from that position. So zooming in, we can see that the compound is making extensive interactions with the pocket. We have multiple hydrogen bonds as indicated with some of these dashed lines. We have van der Waals as well as hydrophobic contacts. And so we were, really curious about why are these so selective? And so to start to, look into this question, we did the sequence alignment for all of the 56 USPs. And there were 4 residues unique to 28 and 25 that really stood out. So we did mutagenesis studies on those 4 that are shown here in the graph. 2 of them, this cysteine and threonine, were larger amino acids than all of the other USPs, where the other 2 had side chains that would present different hydrogen bonding, patterns. And we ultimately saw that mutating any of this, abrogates compound inhibition, really supporting their importance for inhibitor binding. It was also then pretty interesting, I thought, to speculate that, the reason USP28 is potentially highly ligandable, at least as indicated by the large number of hits that we got in our screen, is because it has this large hydrophobic pocket that doesn't exist in other dubs because of the 2 larger amino acids really at these 2 positions, which is breaking the pocket in half. So that then leads to the question of, why is there this large deep cleft on USP28? And that brought us to some, very nice structural work that had been done by, 2 groups, the Kisker and Commander labs, that published in 2019. And what they had shown is that USP25 and USP28 both form dimers. And then in the case of USP25, it can further bring together 2 dimers to form an autoinhibited tetramer. And this is formed by this autoinhibitory motif, this peptide, from one monomer binding into this exact groove where our compound binds in another molecule of the enzyme. So anyways, I thought this was, really interesting that, we found compounds binding this regulatory site, on a dub. And I think moving forward, what, we anticipate seeing and what we're starting to see a little bit is, rather than finding regulatory pockets that already exist, being able to, use these chemical tools really to understand, regulatory, sites and mechanisms that haven't yet been outlined for different deubiquitinating enzymes. And so then just the last slide on the crystal structure, really focused on, how does this compound inhibit enzyme activity, right? It's not kind of binding, in the channel where the C-terminus of ubiquitin approaches the active site. It's more removed from that. And what we believe the answer is that it's causing conformational changes that really prevent the enzyme from coordinating with that C-terminus of ubiquitin and guiding it to the catalytic cysteine. One of those changes is by shifting this alpha-5 helix about 5 angstroms, which then results in this preceding loop becoming disordered. And this loop is really important for coordinating the C-terminal residues of ubiquitin. And then second, we see that this ASA bicycle is locking a couple amino acids into a conformation that is not conducive for, again, the C-terminal chain of ubiquitin getting to the catalytic cysteine. You need this phenyl group to kind of flip up and make space. So there's definitely more enzymology work to do here, but we do think that this is, significantly, allosteric through these conformational changes. And so thus far, the work that I've described on USP7 and USP28 has really allowed us to identify 3 ligandable pockets on DUBs, all of which overlap with the ubiquitin binding site. So this was really exciting. When we first started getting into DUBs, we hoped to find, maybe one pocket that we could leverage. But we had multiple. And furthermore, which was exciting, is that in parallel to our work finding these 3 pockets, other groups, actually it was all companies that were also working in this space, independently found these same pockets on other DUBs targeted by other chemotypes. And so now you take this together, and it really suggests that we're going to be able to leverage each of these pockets to have focus libraries to ultimately be able to access really a wide range of DUBs. It also means, liking a chemical challenge, that it gives us 3 different pockets to think about for rational design approaches. Now, I'm not going to talk about all of those today. I just, very briefly want to highlight, some work building out our first generation library that targeted the catalytic cysteine before pivoting back to some biology. And so these are really our 2 lead compounds for thinking about a focus library that would covalently target the active site, the catalytic cysteine of DUBs. And just looking at these, we could kind of see a pharmacophore model. You need to have a warhead, you need to have a linker, and then you need to have a non-covalent building block. And this really made sense looking at the structure of the DUB. And so here it's USP7 bound by ubiquitinaldehyde in pink and a non-covalent inhibitor in blue. Right, so of course, you target the warhead, you target the catalytic cysteine with your warhead. You then need this narrow linker to traverse this narrow channel where the C-terminus of C-terminus of ubiquitin binds. The 2 C-terminal residues are glycine. So it's really a thin linker. And then you need a non-covalent building block that can interact with these 2 blocking loops. And we anticipated this would be really important for imparting selectivity. So we made a combinatorial library by combining different building blocks, linkers, and warheads. And really focusing on these warheads that seemed to be productive, for targeting DUBs, but that really look different from the warheads you might see in an EGFR or BTK inhibitor. And then after we had our first several hundred compounds synthesized, we needed to decide how to profile them against the enzyme family. And we ultimately landed on using activity-based protein profiling, which we were really excited about because this is profiling against endogenous DUBs. These aren't purified or overexpressed enzymes. So just very briefly, we can take our cell lysate, treat with inhibitor, co-incubate with the dub activity-based probe. This is ubiquitin that will label the catalytic cysteine of dubs that aren't bound by inhibitor. We then can do an affinity enrichment using the biotin handle, and then quantitative mass spectrometry to see which dubs were blocked from labeling, indicating an inhibitory binding event. And here's the data. The darker the red, the more binding that we observe to a particular deubiquitinating enzyme. And so what was really a relief to us was that we have binding events here. This was a first-generation library. You never know what you're going to get. Furthermore, we have some very selective compounds. Actually, equally or if not, better selectivity than, one of our starting points for the library. And of course, as you might expect, there were some promiscuous compounds as well. And so just analyzing this data, we're actually extremely pleased to see that 60% of our compounds were active, and this was when we defined a hit is greater than 50% block of activity-based probe labeling. But more so than that, we saw that almost half of the active compounds were very selective, and this resulted in 21 dubs being targeted by a selective compound. And those 21 dubs are just highlighted with the red dots around this phylogenetic tree, here on the right. And so what I really just want to highlight is this is now spanning, multiple families of dubs. And it's also, targeting many dubs which are understudied. And so this is always one of the, real pros of a target class approach, is it forces you to break away from just focusing on the 2 or 3 most well-studied enzymes in a family, but looking at some of those that are orphan because you end up with a really good, chemical probe or starting point for it. And so you can maybe see that here with VCPIP1, where we had quite a number of very selective hits and almost nothing known about this particular dub. I know there are at least a few chemists in the room who might be like me and prefer to look at structures rather than, bar graphs and tables. So here are structures. And I think what really stands out is that when we start looking at selective hits for a given dub, you can really appreciate how structurally similar they are at each component, the building block, the linker, and the non-covalent binding moiety. But then those are different, from those 3 components that are now selective for a different dub, really showing, I think, that we're getting this molecular recognition that we designed for, with each building block contributing, to the binding of the given dub target. And moreover, this diversity is really giving us excellent, target spectrum where we can get selective hits across many dubs. I won't take you through all the validation, but, we validated these hits in, 4 or 5 orthogonal assays, very high, hit rates, which I think really just speaks to, the quality of the library as well as our primary screening assay. And so moving forward, we're really well poised to move on to the next generation of libraries, similarly to what we've done here. But now that we have these powerful libraries, how are we utilizing them? And so, I think where we really can make and have been able to, now make significant impact is by using these entire libraries in chemical genetic screens. So we essentially have what we refer to as the dub box. So these are all of our library compounds, just plated out, available to look at any phenotype really of interest. And then like any phenotypic screen, right, you have to follow up with target deconvolution and validation. But this is much simpler when you're starting with, compounds that are already annotated and you're focused on just, 100 dubs as a potential relevant target. And so, I really find this type of screening to be a nice complement to your CRISPR screens for a couple reasons. One is you really can look at acute changes. You can look at things that are happening after only a few hours. And second, you can really identify polypharmacology-dependent phenotypes. So phenotypes where maybe you need to inhibit 2 dubs that are very similar to each other, where you're not going to see any, phenotype with knockdown of just an individual dub. And so we've now been able to, utilize this library multiple times in productive screens. We, published our first, showing that USP10 induces, degradation of oncogenic FLT3, but we've now really expanded. And we have several additional, screens I'm very excited about that are in progress. But what I want to highlight on the next couple slides are just, one of the interesting themes that has emerged from these chemical genetic screens, and then talking about, one that is ongoing. And so we've worked a lot, in the chemical genetic screening area with AML experts here at Dana-Farber, Jim Griffin and Ellen Weisberg, specifically looking for DUBs that are stabilizing, mutant enzymes that are found in, subsets of AML patients. And what's been really interesting is, not only, right, have we been able to find DUBs for several different, mutant enzymes. I'm showing, oncogenic FLT3 and mutant JAK2 here, both of which are published stories. But in these and other cases, what we really see is selective degradation of the oncogenic protein of the mutant protein, over wild type. So this is certainly interesting both mechanistically and therapeutically. For example, in the case of JAK2, concurrent inhibition of wild type by the JAK2 inhibitors has been a real clinical challenge. And then mechanistically, we're really curious to start to learn more about phosphorylation and ubiquitination crosstalk, which is probably really the origin of this observed selectivity. And then finally, we've kind of gone into a new direction with phenotypic screens, or these chemical genetic screens, where we're going beyond looking for just a given phenotype and going straight for looking at changes across the entire proteome at the levels of abundance, ubiquitination, as well as phosphorylation. And I'd really like to acknowledge this being enabled by, a tour de force effort from a talented postdoc, Eric Zhu in Jared Marto's lab. But we've now been able to, profile in a data set identical to what I'm showing you here, a small library of about, 20 DUB inhibitors to really start to understand the direct biochemical substrates that change in ubiquitination. What is their fate? Do they get degraded or not? And then seeing some really interesting examples of changes across the phosphoproteome. And as this technology continues to advance, we're really going to be able to put more and more compounds through and, start to, again, move beyond an individual phenotype to just gleaning information about what the DUB does across the proteome. And that includes the good and the bad, where that gives you opportunity, but also where there may be a liability that's going to make you think twice about wanting to, progress a program around that particular DUB. Okay. And so just in the last 5 minutes here, I then, want to shift from talking about inhibitors and talk about different modalities for DUBs, thinking about activators and thinking about DUB recruiters. And so I'm sure many, people here appreciate that the vast majority of drugs, they inhibit. They inhibit enzymes, they degrade enzymes, they inhibit proteins, they block function. And what's been a very difficult pharmacological challenge is increasing levels of the good things. Things like, tumor suppressors, for example. And so we thought that, perhaps in an analogy to PROTACs, DUB tacs could be developed that would recruit a DUB to a protein of interest, deubiquitinate it, and lead to stabilization of the protein. And so that would be for neosubstrates. But you also could think about activators. And activators would then, be able to stabilize an endogenous substrate. So when we were thinking about where to start here, we kind of went back to our favorite DUB, USP7. And what led us back, to our favorite DUB was the need for both activators as well as then potential to include these activators in a DUB tac. So why would we want activators for USP7? So I learned that there's this rare developmental disorder. It's called Howe-Fountain syndrome, and it's defined by a haploinsufficiency of USP7. This was discovered just a few years ago, where a group at Baylor College of Medicine was looking into 6 patients that had similar, dysmorphic physical features, as well as developmental delays, and discovered that they had these chromosomal microdeletions that was leading to the haploinsufficiency of USP7. A couple years later, this Howe-Fountain syndrome foundation has started, and one of their missions is really to find all of the patients with this disease. So they're now up to, several hundred, and finding that they have point mutations spread all throughout the USP7 gene, with many of them clustered in the catalytic domain and at different positions within the catalytic domain. So the assumption, based on the initial identification of haploinsufficiency or loss of one allele, was that these would be inactivating. But addressing that is a question that we've been working on in collaboration with Irina Besanova at UConn Health. And so when we express now USP7 with these point mutations, either catalytic domain or full length, we see that indeed some of them are inactivating, but not all of them. So it's really telling us, there's more work that really needs to be done, especially in increasingly physiologically relevant context, to understand how these different mutations are driving disease. So we went back to some of our high-throughput screening data to see if we could find an activator for USP7. And we could. Here we saw what looked like a very selective inhibitor of USP7, nice activation, no activity across other deubiquitinating enzymes. Synthesize the compound, test, nice dose-responsive activation of USP7, no activity against the other DUBs that we've tested, including USP7, which is very similar. Then did some enzymology to start to understand how this is activating. And what we learned from this Michaelis-Menten kinetics is that this is really a Vmax-driven event. We were able to, in an orthogonal assay, confirm that this is not just any sort of autofluorescence, but in fact we can drive labeling of USP7 by the activity-based probe with addition of our compound. And as we've now just been able to start to test this compound against some of the different disease-relevant mutants, we're very excited to see that indeed this scaffold is able to activate. And we've now been using computational studies to identify the binding site. So we started really by doing some docking and then MD simulations to see if the compound would potentially bind into either of the known ligand binding pockets for USP7. And really what we observed was that it fell out of both pockets. But what was interesting is that if you let the MD simulation continue to go after it fell out of either of the pockets, the known distal or S3 to S5 tunnel, it kind of floated around a little bit, but then in both cases it settled into a new pocket. And it was the same position in both cases. And when we run MD simulation now with a compound in that pocket, it stays put. It seems to be pretty happy in that particular pocket. And then doing NMR chemical shift perturbations where, we can add compound to USP7 and look to see which residues are perturbed. We see those largest chemical shift perturbations right in the binding region of the compound that had been suggested by MD, really supporting the notion that this is in fact where the compound is binding. So then if we take a closer look at that binding pocket, what's actually very interesting is that it overlaps with where the C terminus of USP7 can bind to lead to activation of the enzyme. So this is likely now mimicking the natural mechanism for activating USP7. This is an overlay of where our compound bounds, with the peptide that is known to activate. It's a C-terminal peptide tail. And if we then, kind of mutate residues in this pocket, we can see that it reverses the activity of our compound, really suggesting that indeed this is the correct binding site. And so just to go ahead and wrap up, hopefully I've been able to show you today that we've done a lot of foundational work to establish a DUB platform that now spans custom libraries, medicinal chemistry, purpose-built assays, biochemistry, proteomics, computational studies, cell biology, and that this platform has really turned out to be valuable in generating chemical tools and allowing, a growing knowledge base around DUB regulation, substrates, and phenotypes. But I think we're also entering a really exciting time where we're really now poised to, start to dig into some more detailed mechanistic questions surrounding individual dubs and the target class as a whole, and some, more sophisticated studies really looking at the therapeutic potential. A few areas in particular that we're, focused on is really understanding, this WSP28, where there's so much literature, half of which says it's an oncogene, the other a tumor suppressor. Really now using activators and other ligands for protein stabilization, and some more work, on thinking about the therapeutic potential of USP7 in the clinic. And so with that, I'd really like to acknowledge all of, the current and former members of my lab, the current ones, listed here, but, being in a lab that's really just starting up requires, a lot of perseverance and, hard work, so I'm very appreciative. And we've been, extremely fortunate to work with great collaborators, both here within Dana-Farber as well as external, and hopefully I've acknowledged them throughout the talk, as well as, thankful, for funding to allow us to do this work. So thank you, everybody, for coming today, and I'd be happy to answer any questions.